The ultimate guide to chemical analysis of biomolecules. Explained in minutes. Have you ever wondered what makes up living organisms at the molecular level? Well, today, we're diving into a fascinating experiment that breaks down the chemistry of life. Stick around because, in just a few minutes, you'll understand how scientists analyze biomolecules in living tissues. Step 1. Preparing the sample. The slurry method. To start, we need a living tissue, like a vegetable piece or liver. We grind it into a slurry using a mortar and pestle, while adding trichloroacetic acid, TCA. But why TCA? Because it breaks open the cells and releases their biomolecules, making it easier to analyze them. Step 2. Filtration and separation. We now filter the slurry using a cheesecloth, separating it into two parts. 1 filtrate, also called biomacromolecules, and 2 retentate, also called biomacromolecules. So, what's the difference? Let's break it down. Step 3. The filtrate, acid-soluble pool, the tiny but mighty molecules. The filtrate contains small molecules, ranging from 18 to 800 daltons in molecular weight. These biomacromolecules act as building blocks for life. Here's what we find in the filtrate. Amino acids, the Legos of proteins, simple sugars, monosaccharides like glucose and fructose, nitrogenous bases, the ATGC of DNA and RNA nucleotides and nucleosides, the powerhouses of genetic material, inorganic components, essential minerals and ions. These small molecules are crucial for all biochemical reactions in the body. Step 4. The retentate, acid-insoluble pool, the giants of biology. The retentate contains larger molecules with a molecular weight of over 10,000 daltons. These are biomacromolecules, essential for structure and function in living cells. Proteins, enzymes, structural proteins, hormones, complex sugars, polysaccharides like starch and glycogen, nucleic acids, DNA and RNA, storing genetic information, lipids, the mystery macromolecule. More on this in a second. Why are lipids in the retentate, if they're not biomacromolecules? Wow, great question. Lipids, fats, oils, steroids, don't polymerize like proteins, nucleic acids, or polysaccharides. Their individual molecular weight is less than 800 Day, meaning they should be in the filtrate. Right? Wrong! When the slurry is made, cell membranes break down, and lipids clump into vesicles. These vesicles have an overall molecular weight exceeding 10,000 Day, so they end up in the acid-insoluble fraction. This is why lipids appear in the retentate, even though they're technically not biomacromolecules. Conclusion: the chemistry of life in action. Biomacromolecules. Small molecules. Amino acids, sugars, nucleotides. Biomacromolecules. Large molecules. Proteins, complex sugars, nucleic acids, lipids. Lipids are unique. They're not truly biomacromolecules, but their vesicles push them into the acid-insoluble pool. And that's how scientists break down and analyze biomolecules in living tissues. If you found this helpful, don't forget to like, comment, and subscribe for more amazing biology content. Turn on the notification bell so you don't miss our next video on carbohydrates. The power source of life, on fire.